I'm very intrigued by AI and all this fast development. For instance, in the antibody drug discovery, it's already pretty good for optimization of molecules to use AI. But for discovery in silico design of new antibodies, it's still early days. Big companies particularly, they find it difficult to implement this technology in their workflow. There's already maybe a bit disappointment from higher management that it doesn't go as fast as they wanted to see it go. These innovations can completely change the landscape, I would say. In 2 to 5 years, drug discovery for some programs could be obsolete.
René Hoet is a biopharma R&D leader with over 25-year experience in antibody discovery. He's a former VP biologics at Bayer. He held leadership roles at Genmab, Virginie Biologics, and many other innovative immunotherapy startups. Hey, René. Thank you to be here. And I think during your 25 years of career, you have worked with organizations of all sizes, small and large, and you have seen technologies come and go. But which one, in your experience, has been a transformative technology that really changed the game in drug discovery?
Yeah, it's a very good question. It's also a difficult question because, if you go back, so it's even more than 25 years. It's, I think now 35 years I'm working in drug discovery. It's always easier to see, from development which were 20 years ago than from the ones which I've worked on a few years ago, because it takes time to develop something. But if I think about the technology which actually enabled others to develop drugs, I think my work at Biotech was actually helped a lot of companies to develop drugs. Nowadays, it's actually technology which is well known, a semi-synthetic phage library. And we actually licensed that to over 20 different companies, pharma and biotechs, and used it ourselves. And from that library, there are 5 antibodies now approved, FDA and in the EU. And there's still, this is published in 2005. There's still actually companies working with that library right now, just for one antibody library. At the moment, the hundreds of libraries and technologies, and it has grown, but, it was one of the first antibody libraries that was successful. So that was, from technology-wise. If I think about a company where, let's say, innovative drugs, and I would say I'm most impressed during the years, it would be a different company. It's more, there I would actually choose Genmab, which is not so much technology-wise, but therapeutically-wise. There have been continuously innovation in immunology, biospecifics. They developed their own platform, dual body platform. So they have developed many drugs that are all on the market right now. Although I worked there only for 2 years, I learned a lot there. And also that company, I think, has contributed a lot to the drug discovery in the last 25 years.
Yeah, so you mentioned 2 companies, one I guess was a bit smaller and one a bit larger. Do you see a difference in which these different size company relate to new technologies, whether they might be risk averse to try something, or did you notice anything in how they approach new technologies?
Yeah, Genmab also started small. It's now a big company, but at that time it was not so big. So I think it was always driven by innovation and the drive to succeed, in a product. Biotech was indeed a bit smaller, and it was earlier in new technology development. I would say it's probably easier at a small company to have that drive and passion, and you can influence technologies easier than with a large company. I also worked at Bayer. I learned a lot there, what it takes to develop a drug, but it's more difficult to influence a company of 100,000 people. That's not so easy done. And that's why actually I went, I decided in my career to step back from these big companies, going to smaller biotechs again, and because there you actually have less people, you can do less, but you can directly influence what you want to do.
And you think that in that respect, a smaller company can be more innovative because maybe it's easier to embrace new technologies, or you think that also maybe the let's say the capacity of large company is more enabling in terms of innovation? Or what aspect do you think is more favorable for innovation?
Yeah, it's probably true that most of the smaller companies, biotechs, have more innovation than large pharma companies. The large pharma companies very often, either they buy or they collaborate with the small biotech biotechs to get innovation in their company. So I wouldn't say there is no innovation in bigger companies, but it's more via collaboration very often. Yes, the last few years I worked with a CSO for immuno-oncology drugs, very innovative drugs. That is really innovation, but I cannot tell you yet, if they will be successful because they're still in clinical trial. So it's too early to tell if these concepts will be successful. But it's certainly, highly innovative.
I can definitely imagine that innovation works differently in this different company, maybe both that innovate. But also, you mentioned an innovation in terms of, you work with phage display at DIAX, and that thing was, back in the days, a major improvement. And maybe working on that, what was the key aspect of that innovation that really drove the technology or the field forward?
It's a good question. I mean, at that time a few companies working on phage display libraries. There was Cambridge Antibody Technology. There was MorphoSys as two big companies. Yeah, they were having different approaches, and so we had an approach. So, for instance, Cambridge Antibody Technology, they didn't give access to other companies to develop the technology, and that was a hurdle. There were patents actually blocking many companies to develop it. So what we did in DIAX, we developed a new technology, which actually was to replace PCR, which sounds a bit strange, but that actually was a new technology to make new antibody libraries. And when that was successful, that was a breakthrough because we could license actually this technology to many pharma biotech companies. So it was scientifically, but also, it was, let's say, commercially a kind of success because of that.
Yeah, so I think what you're basically saying, if I understand correctly, is that opening up the technology actually helps putting the technology forward and extracting most value from the technology from multiple parties that might benefit of such a new technology. And I think we discussed also before in previous conversation about the fact that you also have been at the edge of computational innovations in some of the company that you have been working with. So where do you think that specifically at the intersection between indeed biology and computation you have seen the most innovation? What I think what is driving or you have seen experience being the largest innovation at that specific intersection?
Both at DIAX and Bayer, we actually were using quite a bit of automation in our drug discovery, in collecting really at that point already, sequencing CDRs, frameworks, antibody sequence, hundreds of thousands of sequences. At that time, 2000, I'm talking here about DIAX was around 2004, 2005. So this was pretty early on. There was no NGS at that point yet. In Bayer, there was NGS, so that increased the number of sequences. But we started early on with collecting sequences and collecting binding data and cell binding data, affinities, and put them all in the database. At DIAX, we had bioinformaticians that actually worked on a program we called WebPhage. In Bayer, we actually worked with Gene Data as one of the first companies to actually start up making a database platform, which they later expanded and licensed to many pharma and biotech companies. So that's sort of my involvement. I'm for sure not a bioinformatician, but I sort of understand the I would say the major questions which are important. That's a bit beyond what's now going on with AI. I would say that's a different story, but it started with good collection of good data, I would say.
So indeed, so basically what you experience is the increased amount of data and the necessity to be able to organize that data and put it in the right context. And maybe you mentioned AI, and of course, this is a very hot topic. Just what is your take on that? Where do you think AI, it's really improving currently the field, and where you see it going in the near future?
Yeah, I'm very intrigued by AI and all this fast, development. I mean, it goes so fast. If you don't look for a few months, there are already new models and new approaches available. So it actually is very fascinating to see. On the other hand, there is also some people that are already quite disappointed by AI because they expect that AI could do everything. It's probably true with any new technology that there is a huge enthusiasm and then there is some disappointment, and then you get some steady flow of that it's going to be applied and really useful for many people. it is already making a big impact, and it will be much more even in the future. I think it's important to test the different models that are available, collect the data, test different models. At the moment, for instance, in the antibody drug discovery, it's already pretty good for optimization of molecules to use AI. I think most people are convinced that's actually the case. But for discovery in silico design of new antibodies, it's still early days, I would say. In that context, I found it interesting that a colleague of mine, Andrew Bradbury, set up its contest for many companies to evaluate actually different antibodies he had to COVID-19, and he presented the result quite recently, and it was actually interesting. In one hand, it shows that AI works to some aspect. On the other hand, it also shows, compared it to his own experimental data, that most of the AI designed antibodies were not as good as experimental antibodies, and only in a few cases they were a bit better. A few antibodies were a bit better. So there's still a lot to gain there. And what I find also interesting from that study is that there's so many different models, and everybody used different models, and it was not clear which model was best. And and many of the companies used a combination of different models. So it's not so black and white yet.
So do you think also based on that study, I think, and I personally believe that more of this comparison study are necessary to understand where we are currently in the cycle, let's say on the AI cycle of technology development. Do you think that in this cycle we are still climbing the hype, let's say mountain, and we're not yet at the top? Or you think we are reaching the disillusionment moment, if there is a disillusionment moment for AI in biopharma?
Yeah, it's a good question. I don't know yet. I think there is. It's more. It's still in a bit hype phase, I would say. But I think there is also big companies, particularly, they find it difficult to implement this technology in their workflow. There's already a bit maybe a bit disappointment from higher management that it doesn't go as fast as they wanted to see it go. So it's probably there, sort of at the top, and we'll see where it goes. Hopefully, it could also be a technology which is so fastly implemented that there is no big disappointment. But I guess there will be a bit. It's probably also due to that some of these AI companies, they promise to change the world. They say they can design antibodies from silico, and they only have to test 3 antibodies, and they got a picomolar affinity antibody, which I think most people in the field doubt this is correct, and that these type of hypes, they create for people a little bit outside the field the idea that this is all easy to be done, and which is not. And I think a company of ENPICOM, I think, is very, very important to support companies in the development of different AI models and the implementation of it, and to compare these different models. I think that's very important, I would say.
Yeah, and indeed, I think there are many claims out there, quite bold, and I think those have the potential to create some, let's say, disillusionment or some discontent if those don't actually match eventually. So I think maybe indeed we are still climbing a bit that hype mountain, but there is that potential of being disillusioned by it. And regarding this, I think again you have a very long history, and maybe this is a bit provocative question, but have you seen any technology in your career that you had great hope for, but then eventually turned out not to be that great when you started implementing it or you dive into it?
Yes, quite a number, I would say, because you try new things, and you have the intention that this is easily done, and some things don't work out as you hope for. Usually, these things are not published if they don't work at all. I have to tell. So one of these things, which I think I still have good hopes for, there are papers about it, but people still question how efficient it is phenotypic screening, actually. And phenotypic screening, I mean, in biologics, people typically take a target and then do target discovery based on that target and optimize the antibodies. For small molecules, it's actually quite common to use a phenotypic screening. So they start up with a particular assay, and then they screen the whole big small molecule library for certain hits, and they find some initial hits which they optimize. So they start with the function first. With biologics, it's the other way around, which has the disadvantage that, or the advantage that you know where you're going, but the disadvantage that you get at maximum what you expect. So you don't get new things. Real innovation comes from things that you don't expect. And that I feel that phenotypic screening could actually make a difference. For example, if you start off with cancer cells and you select on certain cancer cells, or certain, and you deplete on normal cells, and then you screen for some function of those to inhibit proliferation, or then you can find quite novel things. For example, novel glycosylations on cancer cells. It's known that they are there. So you could select for a novel glycosylation site, which is only on cancer cells and not on normal cells. Or so there could be, and people have been successful in that approach. So there are even antibodies found early on, and one of them is for example alemtuzumab. That's an antibody to CAMPAS. It's called CAMPAS. It's an antibody which was validated in the clinic already in 1984, so a long time ago, without that the target was known. And so only in 1991 the target was identified. So there are historical cases where people actually found that. I would say the last 20 years this is limited. There are a few companies that actually are doing this phenotypic screening. For example, BioInvent, they have this platform function first. They screen antibodies first for function and then characterize the targets. And they have some antibodies in clinical trial. And I think the advantage of this function first is also that you can easily incorporate the basis of the patient, use patient data. I think that's for me the important step. If you start with a target, then you only at a later stage screen typically for the function. You find first antibodies that bind and maybe the affinity, and then you go to function, and then in the end you look at what impact that has, and then you're typically limited to a few only. Well, if you start off with antibodies, first select for example, an agonistic function, high throughput, and then you're going to the next phase, then you come from a completely different angle. So I still have good hope for that. New technologies might help here. There's new microfluidic technologies where you can now screen millions of cells. So if you can screen millions of cells for functional activity, then you might be able to come there faster, because that was one of the limitations. It was very difficult to screen these millions of cells for a functional activity, particularly if you think about, for example, B cells, that are not immortalized, they don't live a long time. So you have to do this screening in a very short time window to be able to successful. We worked on this and are still working on that in Fair Journey Biologics with the technology mammalian display. There you make a copy of the B cells in mammalian cells. You incorporate that by stable cloning, and then screening is set up for for functional screening. That's still in a development stage. You can find easily binders to a molecule, but the idea is in the future this will be possible also for functional activity. And I think that might help also to find some novel things.
And you think that the main reason why, for instance, phenotypic screening is being applied successfully, or is being applied more successfully to small molecule rather than biologics and antibodies, is mostly because there was a limiting factor in terms of the number of antibodies that could be screened, or there were also other factors that you imagine contributed to not that being, such maybe more widespread technology for finding new drugs?
If you start with a cell which has 10,000 different targets and they're all different glycosylation, and you apply an antibody library of 10 to the 10 variants, you need to be able to screen that in a sufficient way. And the display technologies typically use not full antibody molecules, only fragments. And the fragments you can actually not test that well for function. So for instance, FAP or single chain or single domain typically are not enough to create an agonistic activity. So you need the whole molecule to make. And there is not that many display systems, except mammalian display, I would say, and yeast display to some extent, where you can actually make the full molecules and make them available and make very large libraries that you can then screen for activity. B cells is one of the possibilities, single B cell screening, which has been developed the last 10 years or so extensively. And there you could also try to do this, or actually go from the B cells directly to the mammalian cells and then screen for activity. So I still have good hopes that these type of technologies will help to create more, let's say, functional activity. Another area I think where this phenotypic screening is needed and will help quite a bit is in these bispecific and multispecific antibodies, because there you put activities together, 2, sometimes 3 activities, with the idea that you want to have a functional activities. And if you have 100 antibodies to one and 100 to another one, then you already have 10,000 combinations. And how do you test all these things separately? Typically, it's too much to test in producing all these proteins. And so it would be nice if you could do on a cellular level, make these molecules in the library and select them in advance, before doing all that work. So I think there it could also help quite a bit.
And do you think that maybe the tendency for large pharma to go after the same targets and therefore have a more target screening approach rather than that phenotypic broader screen that might actually unravel a new mechanism has something to do with that tendency of choosing maybe more a targeted approach for antibodies rather than a phenotypical, more broader screening? There is a political, let's say, take on that as well?
I would say particularly in large pharma companies, that there is a tendency to not take too many risks, try to reduce risk. That's more or less, to be successful with as little as risk as possible. For example, I think you mentioned this already, there's so many antibodies already developed to CD20, 20 different ones, I think, already in the clinic. HER2, I looked it up, it's 40 different antibodies in the clinic. PD1, over 50 different antibodies developed by different companies in the clinic. So not just for this discovery. All with the idea, one is successful, so we make another one and we try a different combination, which I think is, in my view, a bit waste of energy and time. And it would be better placed by trying to find something new. And there are examples also that something new completely changes the world. This is not for biologics, but I'm just thinking on the new drugs for obesity and diabetes, Ozempic, and that actually changed completely the drug discovery for these type of molecules. And for this year, I saw that 4 of the 10 best-selling drugs are now these drugs. 60 billion sales per year, and that's in a matter of 3 years, 2-3 years. So these complete innovations can completely change the landscape, I would say.
And do you see maybe the possibility to combine actually the phenotypical screening with the targeted screening? Because I totally see how phenotypical screening can unravel new targets, perhaps, and open up completely different opportunities for patients and also markets. So do you see the possibility to combine the two?
Yes, I think it is possible to combine them, and I think it is, it's important for a phenotypic screening to have a very robust screening assay, I would say. Ideally, have materials available from the patients there, and then unravel, by the target pretty early in the discovery process. There are systems available, and we use that in Bayer also to identify the targets, like technology developed by Retrogenics, which is now taken over by Charles River, which you can screen against all human targets, 5,000-6,000 targets. You can screen your antibodies. So once you find an interesting antibody with activities, you can quickly go to the target, and then, because this might not be the best antibody, but then you have a starting point and you can use a traditional way to further optimize and make more antibodies to such a target. So I definitely think there is a possibility to combine things. And perhaps AI in the future could also combine a bit earlier, maybe even in finding new mechanisms or combinations of targets. That's an area I think, which is also very interesting, to find new things there.
Yeah, absolutely. So that's indeed one of the questions that I wanted to ask you, whether the fact that AI might help targeted screening will make maybe that even maybe a more sought choice for pharma, because maybe they can actually identify via AI novel target with that technology. So maybe that's one question. And the other that I wanted to ask you is, do you think that AI might replace phenotypical screening at some point, and you would be able to do the same type of screening with AI at some point in the future?
I mean, phenotypic screening you would use to create innovation in it, which is probably difficult to predict, at least at the moment. Maybe in the future it's different, but so I don't see that immediately replaced by AI. But AI can certainly enable it and further develop it. And the other question you had, sorry, I missed. I started with the second one.
No, exactly. The second one was maybe more provocative, right? So about replacing completely phenotypical screening. But maybe if we look at the target discovery approach and the fact that maybe AI can help finding new targets, and hence, replacing a bit the idea of phenotypical screening as in finding new mechanism, because that could actually be achieved via AI, and then reverting back on a target discovery approach. Would you see that as a concrete possibility, or you think you're doubtful?
No, I do see it as a possibility, concrete possibility. A company where this is already actually done is a successful company, Alchemap. I don't know if you know that one, which is they are using actually patient data from resilient patients that actually sort of recovered from disease, and they do this in the area of neurology diseases. And they use antibodies from these patients, the data from the diversity of these antibodies, compare that with the patient that don't have this inhibition for the disease, and then they found actually with AI they found identified antibodies that are unique for these patient subset, and then identified also the target. And a few of those antibodies are now in clinical trial. So I think that's actually a very nice combination of using AI and the type of phenotypic screening, different than I mentioned before on cells, but here it's using directly patient data.
I think that's very interesting. And where do you see, let's say, the low-hanging fruit of AI in drug discovery? Because I've been, to conferences and places, and AI has been adopted across the spectrum, from both improving molecules, but also in the lab, also in the clinical aspects, extracting data from patients' reports and so on. So where do you see the most, let's say, important or the most impactful innovation at the moment?
I wouldn't be able to answer the things on the clinical trial piece because I'm not really an expert in that one. I see that can have a huge impact because it's such a long period and such an important part of the drug discovery, to design clinical trials well for the right patients and look at biomarkers. And that's later on in the discovery, but can have a major impact. For the early part, I would say AI, if it can actually design new drugs based on a target structure or structure prediction to a certain epitope, there it could make a huge impact. And perhaps in 2 to 5 years, drug discovery for some programs could be obsolete because we know the structure of a target or domain, and we can design antibodies that bind to those domain as a starting point and optimize them. So that would be a fantastic sort of step forward, from silico design based on structures to be done. So I think that's a dream that many people have. I think it can come true, but it will take a few years.
Coming back to the mammalian display and these technologies that you also help other companies implement and adopt. And of course, in those companies were probably innovation that they were trying to achieve. What are the obstacles that you face in trying to implement those innovations? So are specific obstacles that you would say you would have to face in implementing innovation in smaller or larger companies?
Mammalian display is a quite sophisticated technology, because this is stable integration in the genome, and you have to select huge number of cells to make large libraries. So I wouldn't say you can implement this easily in any company. So there need to be some steps there. Molecular biology, cell biology needs to be quite broad established. For example, the Iontas, which is basically now part of Journey Biologics, they developed the technology and licensed this to BMS. They actually set up the transfer process, and they were able to implement this actually pretty well. But that's a big pharma company. I wouldn't say this is easy done by every biotech company.
So indeed, so you require a specific set of expertises to do so. But do you think that those are mostly technological issues that needs to be overcome or adopted, or there are also other aspects in implementing innovation, perhaps with management or other aspect like that you think you experience to be critical to implement innovation?
Innovation needs, the drive to succeed. I would say that's sort of the passion to succeed. That's very, very important, and that passion should come from all levels. Investment for equipment, for example, for a mammalian display, you need very sophisticated fax sorters machines. So you need them. So that's an investment needed. Of course, you can try to set up your own technology. That will take even more time. Or you can license. So you need that financial piece. You need to be in place, and then you need good scientists to implement, and that they have time enough to implement and validate. And there should be no, let's say, unrealistic expectations. That's also important. I think my experience, at least with large pharma companies, they can be very enthusiastic initially, but then they want to see immediately also the results. So there's only a small time window you have to succeed.
So there's definitely that limitation in time. But indeed, you mentioned, for instance, the case of a mammalian display, you need to bring together different expertises. You need to bring together, like cell and molecular biology and, bring these two expertises together. Now, of course, with AI and more computational, there is yet another aspect, the computational biologist, and let's call it the computer people joining the crowd. Do you think there is also a cultural aspect to be considered, like how to make these different people talk together and brought together as a key component of innovation?
Absolutely. I think silos are actually the worst scenarios, and it is more difficult. The bigger the company is, the more difficult it is actually to prevent silos. In my career, I always have to try to build bridges between different expertise and structures. I think it's really important because that's the only way to be successful in developing a drug. The companies that are successful, an example of a company that I not worked for, but which is also very successful, is a company, Argenix, in a Dutch company, Belgium, in Ghent. We worked together as buyer with them, and they did an early discovery program for us. They were not successful, but we saw with all the scientists, the whole company was so motivated and so driven to succeed that, I'm actually not surprised that the company is now a very successful biotech pharma company. So it needs a drive, in my view, and from upper management to really the scientists, really to succeed.
So there is the drive, and I totally see that the whole organization need to be aligned towards that objective. And maybe when it comes indeed to the part about bringing the computation into the drug discovery, do you see also the necessity to have or to develop specific skills for that? Or, let's suppose that you would need to mentor a junior scientist coming in drug discovery. What would you recommend them to, be proficient at, or in addition to his own very peculiar expertise for be successful in the future?
In big companies, you have these professional development career programs for scientists, not just on the science base, but also on management skills and leading a group. That's a big thing if you come from academia and you suddenly are responsible for managing a group. So those things you have to learn. I really personally, I like teaching young people. I'm also, teaching at the university in Maastricht. Try to, to show to them what it takes to develop a new drug from early start, from the target to get an antibody approved. And very often people come then afterwards to me, these are master students, and say, we would like to, what is it to work at a company? Because those people are usually have only academic experience. We would like to try. I said, yeah, please go ahead. Try out. Work there for. Try to get a interim period to work for 3 to 6 months and try to understand if this is what you want to do or you want to stay in academia, because that is still a hurdle for people coming from academia, to make that choice early on in their career.
What do you think is the role of academia in terms of innovation in industry? You would consider a key role, or what's your experience there?
I think academia plays a key role in bringing up new concepts in my view, and new ideas. It's very important that there is a kind of a bridge between academia and then industry, because many of these ideas they stay in academia because they're not far enough developed so that biotechs or pharma can pick that up. So that, I think, is a hurdle which is still there in academia. I do think that there's lots of new innovation there. Making the bridge to applications is more difficult. Some of these academia try it themselves, to commercialize. If they don't start an official company, I think that's probably not a good idea. You need really to make a choice there, what you want to do, and then fully go for it. And there are people, of course, that successfully spin off a company from university and developed it into a company. The successes for like GenMap and Argenix and Neplings, those are basically started by academic founders that actually started those companies.
And you mentioned that one of the key elements to success is indeed embracing fully, the going to the industry and therefore maybe leaving academia behind. Do you think there are other key elements to transfer the opportunity from academia to industry, especially for the people, or are there some lessons learned from your experience regarding that?
Yeah, so early in my career, I was working for a company, Target Quest, which was founded by Henny Hogenboom, one of the pioneers in phage display. And he had also an academic group. And he managed to convince the university that actually he could set up next to his academic career also a company. That was a difficult process, I can tell you, because they then the university saw that the company was successful, and then this started in a legal fight, and which was not good. So I think that was early in biotechs. It's now probably better arranged in academia. But yes, it is a hurdle, I would say, to develop technologies from a academia to a biotech or pharma environment.
Is there a question you would have wanted me to ask you that I didn't, and you would like to answer?
So I ended up in industry, but actually early on in my career, I had to make a choice. Actually, I thought I would always be an academic in my whole career early on. So then I got a job in Maastricht, the University of Maastricht, where I was 50% working as an academic and 50% for this company Target Quest. And after a year, I got a permanent job offered at the university. But I was so fascinated by a small biotech company, because we started, with 1-2 persons, and within 1 year we were 10 people, just financed by CRO work we were doing. That actually I chose for the company, and I never regretted that actually. Some people have an understanding of companies that you don't share, or everything is secret and you cannot share things, and that's a wrong thing in my view. I think there is within the company there is also a lot of innovation, and so that's maybe my personal thing for my career. I do like university and academia because I'm also part-time professor there, at the University of Maastricht, but I think in real developing new products is very fascinating, and yes, I really enjoyed that path in my career.
I think that's a great story, and I think a lot of people that made the switch from academia to industry can relate to that. And also in my own personal experience, when I was a postdoc, at that moment, it for me was clear that I wanted to change my career path, and I wanted to go to industry. Actually, I wanted to start an entrepreneurship kind of journey. And I clearly remember that moment at that point in time in a similar way in which you are recalling that moment for yourself, in which you wanted to experience more of that biotech. Why do you think that people hesitate to do that? Is just because they think there is not going to be enough research? What do you think is the issue there?
They don't know so much about it. In studies, typically, at least at universities, they are exposed mainly to the university. And therefore, I think it's also important that people like you and I, for example, also tell a little bit about what happens in industry and motivate people to take those steps.
There is life after academia,
exactly,
in a way. Yeah, no, I can totally relate to that. And indeed, make sure that they understand that a lot of great research actually is done in industry. And of course, there are sometimes different goals, sometimes different timelines, but it's a great place to see and execute on innovation. So that's, I think, is an important message to give to all the younger people that are asking for the same questions. I like often to terminate this kind of conversation with an open question, a bit challenging, to see what people think about the present, but also about the future. And this is based on a contrarian question. Peter Thiel often asks that question, which basically says, what do you believe to be an important truth in the field of drug discovery, which is your field, that you believe to be indeed really true, but your peer and others would disagree with you?
My drive is innovation and new concept and new products. And in discovery, I usually choose the new steps, the new areas. And for example, phenotypic screening, we already talked about this, so I don't want to tell too much about it, but it is an area where you can find innovation. I can tell you, although some of my bio colleagues liked it, in a big pharma company, this is very difficult to actually get support for. So most of my colleagues didn't agree with me and said, let's do just the targeted approach. We know what we have after a year. It's about risk taking a bit, yeah. So I think that's where I differ probably a bit from most people in the field.
We should be taking a bit more risk. That's your final comment.
Yes.
Well, René, it has been a pleasure to talk with you and have this conversation. I think we learned a lot, and you had a great career. So thanks for joining us. Thanks for joining the show.
Thanks very much for giving me the opportunity. I very much like the conversation. Thank you.
Absolutely.